The treatment of obesity has changed dramatically in recent years. Drugs such as Ozempic, Wegovy and Mounjaro have demonstrated that substantial weight loss can be achieved by targeting hormones that regulate appetite and metabolism. But researchers have continued to push this approach even further. An experimental drug called Retatrutide has produced some of the largest weight reductions ever achieved with a medicine. In clinical trials, many participants lost more than 20% of their body weight, approaching results previously seen only after bariatric surgery. Unlike earlier drugs, Retatrutide does not mimic just one or two hormones. It combines the actions of 3 different hormonal pathways into a single molecule, simultaneously influencing appetite, glucose regulation, and energy expenditure. In this video, we will explore how Retatrutide works, how it was engineered, and why it may represent one of the most sophisticated metabolic medicines ever developed. Following a meal, the body does not release one metabolic hormone in isolation. It releases a coordinated cocktail of peptides that act together in an integrated way to control appetite, blood glucose, and energy expenditure. Retatrutide was designed to reproduce part of that integrated physiological response. Let's start with what these hormones are and where they come from. GLP-1 and glucagon have very different physiological effects, but they share a common origin. Both hormones are produced from a 160 amino acid precursor peptide called proglucagon. Proglucagon is expressed in several different cell types, including alpha cells of the pancreatic islets, endocrine L cells of the small and large intestine, and certain neurons within the brainstem. The proglucagon molecule contains the amino acid sequences of several smaller peptide hormones embedded within its structure, including both glucagon and GLP-1. These hormones are released when specialized enzymes known as prohormone convertases cleave the precursor at specific sites. The particular convertase enzyme expressed depends on the cell type. As a result, the same proglucagon molecule can be processed in different ways in different tissues. In pancreatic alpha cells, proglucagon is predominantly cleaved to produce glucagon, together with several other peptide fragments. In contrast, intestinal L cells and certain neurons in the brain process proglucagon differently, producing GLP-1 and GLP-2 instead. Thus, although glucagon and GLP-1 perform very different functions within the body, they are in fact closely related hormones, generated from the same precursor molecule by tissue-specific processing. Understanding this relationship provides the first clue as to why scientists became interested in targeting both pathways simultaneously in drugs such as Retatrutide. GIP or gastric inhibitory peptide is also known as glucose-dependent insulinotropic polypeptide. It is made in much the same way as glucagon-like peptide, but it is synthesized in K cells of the duodenum and jejunum. GIP is made from a 153 amino acid pre-pro hormone precursor that is distinct from pro-glucagon. It is called pro-GIP and contains the amino acid sequence of GIP between residues 51 and 93. A prohormone convertase cleaves either side of the GIP sequence to release the mature GIP peptide from its precursor. GLP-1 and GIP are known as incretin hormones because they are released in response to food ingestion and help regulate blood glucose. Glucagon is not an incretin hormone, but works alongside them as a key regulator of energy metabolism. Together, these three hormones exert complementary effects on the pancreas, brain, gastrointestinal tract, liver, and adipose tissue. GLP-1 stimulates insulin secretion from pancreatic beta cells while suppressing glucagon secretion from alpha cells. It also promotes satiety through actions on the brain and slows gastric emptying, causing food to remain in the stomach for longer. Together, these effects improve glucose control and reduce food intake. Like GLP-1, GIP stimulates insulin secretion following a meal, but it also stimulates glucagon secretion. It acts on the adipose tissue, where it promotes the uptake and storage of fat and improves insulin sensitivity. In addition, GIP reduces gastric acid secretion, which slows digestion and contributes to feelings of satiety. Glucagon has traditionally been viewed as the physiological counterpart of insulin. When blood glucose falls, glucagon stimulates the liver to release stored glucose into the circulation. However, glucagon also has important effects on energy balance. It promotes the breakdown of stored fuels and increases energy expenditure, allowing the body to burn more calories. These actions have made glucagon receptors an attractive target for the development of new obesity therapies. Thus, while GLP-1 primarily reduces food intake, GIP enhances the metabolic response to nutrients and glucagon increases energy expenditure. By combining the actions of all three hormones in a single molecule, Retatrutide aims to achieve greater weight loss than earlier therapies. These are the amino acid sequences of GLP-1, glucagon, and GIP, aligned from the amino terminus on the left to the carboxyl terminus. At first glance, they appear to be three different peptides, but they're actually members of the same evolutionary family. If you scan across the rows, many of the residues are identical or closely related, as highlighted by the coloured boxes. Sequence homology is particularly pronounced near the amino terminus, which is especially important for receptor activation, although residues throughout the peptide contribute to receptor binding and specificity. This evolutionary relationship is important because their receptors belong to the same family of class B G protein coupled receptors and recognise many of the same structural features. This means that medicinal chemists don't have to link three separate hormones together. Instead, they can engineer a single peptide that preserves the key receptor interacting features of all three. Retatrutide is a single engineered peptide that takes advantage of this shared structural language. It was developed from a GIP-based backbone and, as indicated by the coloured boxes, retains much of the GIP sequence and consequently preserves activity at the GIP receptor. Because GIP itself shares substantial homology with both GLP-1 and glucagon, with the conserved residues highlighted in yellow, it proved possible to increase activity at the GLP-1 and glucagon receptors while preserving GIP receptor activity. This was done by systematically modifying selected amino acids within the sequence. individual substitutions were introduced and tested to optimize the balance of activity across all three receptors. For example, replacement of position 13 with the non-natural amino acid alpha-methylleucine favoured a peptide conformation associated with enhanced glucagon and GIP receptor activity. Substitution of alpha-amino isobutyric acid at position 20 further enhanced GIP receptor activity. Through this iterative process, a single peptide was created that acts as an agonist at the GIP, GLP-1 and glucagon receptors. Having optimized receptor activity, further structural modifications were introduced to improve the pharmacokinetic properties of the peptide. Amidation of the carboxyl terminus and substitution of alpha-amino isobutyric acid at position 2 protect the molecule against cleavage by dipeptidyl peptidase 4, thereby increasing its metabolic stability. Finally, attachment of a C20 fatty diacid side chain to the lysine residue at position 17 promotes reversible binding to albumin, reducing clearance and extending the plasma half-life to several days. Together, these modifications transform what would otherwise be a short-lived peptide hormone into a drug that can be administered by subcutaneous injection once each week. GLP-1, GIP and glucagon each act through their own distinct G-protein coupled receptor. Although the receptors are different, they all couple predominantly to the GS protein. When a hormone binds to its receptor, the G protein is activated, and the G-alpha S subunit stimulates the enzyme adenyl cyclase, which converts ATP into the second messenger, cyclic AMP. Cyclic AMP then activates several downstream signaling pathways, principally protein kinase A, exchange protein activated by cyclic AMP, and cyclic nucleotide gated ion channels. The distribution of these receptors varies between tissues, and the relative importance of each signaling pathway also differs depending on the target organ. For these three hormones, protein kinase A is largely involved in regulating gene expression and cellular metabolism, whereas EPAC plays a key role in secretion. The effects on ion channels influence beta cell excitability, promote calcium influx, and ultimately enhance insulin secretion. Retatrutide is designed to activate all three of these receptors simultaneously. By combining GLP-1, GIP, and glucagon receptor signaling, it amplifies cyclic AMP-mediated pathways across multiple tissues. The combined effect is improved glycemic control together with substantial weight loss. This novel receptor pharmacology is impressive, but ultimately, the value of any new therapy depends on what it achieves in patients. This graph summarises the results of a Phase 2 clinical trial reported in the New England Journal of Medicine. Adults with obesity, or overweight with weight-related complications, received weekly subcutaneous injections of retatrutide at different doses for 48 weeks. Several features stand out. First, there is a clear dose-response relationship, with progressively greater weight loss at higher doses. Second, the reduction in body weight is both substantial and sustained. Participants receiving the highest dose lost on average approximately 24% of their baseline body weight after 48 weeks. These findings support the underlying biological rationale. By reducing food intake through GLP-1 signalling, enhancing the metabolic response to nutrients through GIP, and increasing energy expenditure through glucagon receptor activation, Retatrutide produces a degree of sustained weight reduction that begins to approach that previously associated mainly with bariatric surgery. The effects were not confined to weight loss. Participants also showed improvements in a range of cardiometabolic risk factors, including blood pressure, glycated hemoglobin or HbA1c, fasting blood glucose, insulin, and lipid profiles. Many participants with pre-diabetes reverted to normal glycemia during treatment. So the drug appears to have broad effects on metabolic health. The safety profile was also reassuring. As with other incretin-based therapies, the most common adverse effects were gastrointestinal, including nausea, vomiting, diarrhea, and constipation. They were generally mild to moderate in severity and tended to improve with continued treatment. The impressive effects on body weight are only part of the story. Because retatrutide was designed to influence glucose homeostasis as well as appetite and energy expenditure, it is equally important to examine its effects in diabetes. This graph shows results from a phase 3 trial investigating retatrutide in people with type 2 diabetes, which has just been reported in The Lancet. It is apparent that glycated hemoglobin, HbA1c, fell progressively over the course of retatrutide treatment compared with placebo. This indicates a substantial and sustained improvement in long-term blood glucose control. An interesting feature of the results is the lower doses appear to achieve glycemic control comparable to the highest dose, although the same patients showed dose-dependent weight loss similar to that found in the obesity study. This implies that glycemic control may be achieved at lower doses than required for weight loss. The study also demonstrated similar improvements in cardiometabolic risk factors and a safety profile broadly consistent with that observed in the obesity trial. Taken together, the findings from these two trials suggest that retatrutide is not simply a weight loss medication that happens to lower blood glucose, but a comprehensive metabolic therapy capable of addressing several key features of metabolic disease simultaneously. Retatrutide represents the next step in the evolution of metabolic therapies. By integrating 3 hormonal pathways into a single engineered peptide, it moves beyond simply reducing appetite and begins to reshape multiple aspects of energy balance simultaneously. However, several important questions remain. The long-term safety of sustained glucagon receptor activation is still being evaluated, and the durability of weight loss beyond the trial period is not yet fully understood. In addition, as with other highly effective weight loss therapies, considerations such as cost and accessibility, as well as long-term adherence, will play a major role in determining its real-world impact. It is also important to emphasize that retatrutide is still an experimental drug. Ongoing phase 3 trials will determine whether these early results translate into safe and effective treatment at a population level. Nevertheless, the results so far are striking. Retatrutide demonstrates that by carefully combining multiple hormonal signals, it is possible to achieve levels of weight loss and metabolic improvement that were once thought to require surgery. More broadly, it highlights a shift in how we approach metabolic disease, not as a single pathway to be targeted, but as a complex system that can be precisely modulated through rational molecular design.